This episode of GT the Podcast is supported by Alcon.
This is Ike Ahmed.
And I'm Arsham Shaybani.
And we want to welcome you to GT the Podcast.
We're bringing this to you together with BMC and Glaucoma Today,
to offer audible insights into current topics in glaucoma care.
presented by the authors of our latest, most read GT articles.
Check it out.
Welcome to GT the Podcast. In this episode, Dr. Francesca Cordeiro recaps her article, DARC, From Bench to Bedside, which was featured in the March-April issue of Glaucoma Today. In this article, Dr. Cordeiro poses that the detection of apoptosis and cell stress holds promise for detecting neurodegenerative disease ahead of existing endpoints. She reviews a novel retinal biomarker, the detection of apoptosing retinal cells, or DARC, and its potential to function as a surrogate endpoint in glaucoma and other conditions. Because retinal ganglion cell apoptosis occurs in the early stages of glaucoma, apoptosis identification may be key to earlier treatment of the disease and prevention of vision loss in glaucomatous eyes. More on this from Dr. Cordeiro.
The management of chronic neurodegenerative diseases such as glaucoma, AMD, Alzheimer's, and Parkinson's disease, as also stroke, is complicated by a similar set of challenges. All these diseases cause significant disability, owing in part to their late diagnosis, the absence of appropriate screening tests, ineffective monitoring, and a lack of early clinical endpoints. Patients with neurodegenerative diseases progress slowly and are followed over long periods of time. Reliable indicators are therefore needed to measure disease progression and efficacy of treatment. In glaucoma, the gold standard for diagnosis is still visual field testing. Visual field defects, however, can take years to develop, by which point at least 30% of the eye's retinal ganglion cells may be lost. This inability to identify early disease is estimated to translate to about a 10-year delay in the diagnosis of glaucoma. Diagnostic delays affect patients with other neurodegenerative diseases as well. Parkinson's disease is diagnosed only once a patient presents with symptoms, by which point 70% of dopaminergic receptors have typically already been lost. With Alzheimer's disease, the time between the start of the disease process and the development of memory loss is reported to be about 20 years. The detection of apoptosis and cell stress is a promising strategy for detecting neurodegenerative disease before irreversible damage occurs, and also for monitoring the treatment response ahead of existing endpoints. Here we review a novel retinal biomarker, the detection of apoptosing retinal cells, or DARC, and look at the potential for this to function as a surrogate endpoint in glaucoma and other conditions. A biomarker is a characteristic that can be objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacologic responses to a therapeutic intervention. A clinical outcome is defined as a characteristic or variable that reflects how a patient feels, functions, or survives. A surrogate marker is used as a predictor of a clinical outcome. For example, blood pressure is the surrogate of stroke, which is the true clinical endpoint. In ophthalmology, we are looking for surrogate markers of visual function loss. We know that retinal ganglion cell apoptosis is the earliest event in the development of glaucoma, and peak apoptosis occurs in the early stages of disease. Apoptosis identification may therefore be key to early treatment and prevention of vision loss in glaucoma. In early apoptosis and cell stress, the cell membrane changes its structure. Phosphatidylserine moves from the inside to the outside of the cell, serving as the cell's eat-me signal for resident phagocytes. This process of phosphatidylserine externalization is reversible, allowing sick or stressed cells to return to a healthy state if appropriately targeted by treatment. DARC uses a fluorescent-labeled annexin 5 protein administered via intravenous injection to identify apoptosis. Annexin 5 binds to exposed phosphatidylserine with high affinity, and individually labeled cells are visualized in the retina as white spots on retinal images, which are captured with standard imaging equipment. DARC technology allows the opportunity to not only monitor disease activity, but also treatment efficacy, and this can be seen by a reduction in the number of white spots, a reduction in the DARC count. My colleagues and I have worked to validate this concept and show that DARC can be used as a clinical biomarker to determine disease activity. Preclinically, we evaluated the use of DARC in several models of disease. Our initial objective was to show that DARC would do what we predicted, so the first endpoint assessed was retinal ganglion cell death. Using whole retinal amount immunostaining and an automated script for retinal ganglion cell counting, we were able to determine that the DARC count occurring in vivo correlated with the endpoint of histological retinal ganglion cell loss. In a study using a partial optic nerve transection animal model, DARC was used to compare the direct application of Schwann cells with intravitreal Schwann cell delivery and evaluate the effects of these approaches on retinal ganglion cell apoptosis and loss. With DARC, we were able to show the direct application of Schwann cells prolonged retinal ganglion cell survival. We found that we could visualize retinal ganglion cell apoptosis with DARC in vivo, and that the analysis of the DARC count in vivo corroborated findings of the retinal ganglion cell count in ex vivo histology. In a study of a rat model of ocular hypertension, we showed that the DARC count was significantly reduced by treatment with coenzyme Q10, and this translated to increased retinal ganglion cell survival, thereby validating DARC as a surrogate of retinal ganglion cell counts. In another investigation, topical nerve growth factor was assessed to be neuroprotective in a model of optic nerve transection using DARC and retinal ganglion cell counts. The results of the retinal ganglion cell density corroborated indications of retinal ganglion cells provided in advance by in vivo DARC imaging. With these validations, we then went a step further to assess whether DARC was predictive of changes in the brain. We found that in a rotenone-induced animal model of Parkinson's disease, DARC could detect the loss of dopaminergic cells in the eye 90 days before it could be detected in the brain. Additionally, in a triple transgenic model of Alzheimer's disease, DARC was predictive of a therapeutic effect of a novel curcumin nanoparticle formulation, which is a derivative of turmeric, before the effect could be observed in the brain. In a rabbit model of wet macular degeneration, human VEGF was delivered to initiate vessel leakage and cause angiogenesis. Dark spots were observed along the vascular streak only in the eyes with the VEGF treatment, illustrating single spot indicating endothelial cell stress. When these eyes were treated with an anti-VEGF agent, a reduction in the DARC count was observed. This investigation further validated DARC as a biomarker of angiogenesis-related retinal diseases such as wet macular degeneration and diabetic eye disease. Moving DARC into clinical trials has involved significant collaboration between the Wellcome Trust, University College London, Imperial College Ophthalmic Research Group, and the Western Eye Hospital. A large team and generous funding have allowed us to take DARC from the preclinical stage to phase 1 and phase 2 clinical trials. The phase 1 clinical trial represented the first time that fluorescently labelled annexin, and we call this annexin 776, was given to a human. So the primary objective was to evaluate the safety and tolerability of annexin 776 for DARC. The secondary objective was to determine the efficacy of DARC and the difference in the DARC count between healthy control eyes and glaucomatous eyes. A total of 16 patients were included in this phase 1 trial, 8 of whom had glaucoma and 8 without. The findings in the glaucoma cohort, although small, were encouraging and also suggested that DARC could be used to predict which patients were going to progress. No adverse events occurred in the phase 1 clinical trial, so we increased the size of the study cohort for the phase 2 clinical trial. This study included 73 patients with glaucoma, macular degeneration, optic neuritis, and Down syndrome, as well as 40 healthy control. All patients in the study received intravenous administration of 0.4 milligrams of annexin 776, and central images of the optic nerve, head, and macula were recorded at baseline, 15 minutes, 2 hours, and 4 hours after administration. 5 masked observers reviewed 906 anonymized retinal images that were randomly displayed on the same computer and under the same lighting conditions. We had assumed it would be easy to count these bright spots of DARC. However, we found the observers had disagreement amongst themselves, and in hindsight, this is not too surprising since clinicians tend not to agree at the best of times, but particularly on something new, such as how to define a DARC spot. We then decided to explore the use of machine learning with a convolutional neural network. Anonymized dark images were acquired from the 40 healthy control patients and also from those patients with glaucoma who were enrolled in this phase 2 trial. The CNN-aided algorithm was trained and validated using manual counts from control patients and then tested on the glaucomatous eye. So the ground truth was where 2 or more of the 5 manual observers agreed a spot was a spot. We found that the algorithm had 97% accuracy, 91.1% sensitivity, and 97.1% specificity in terms of spot detection when compared with the manual grading of the 50% of the eyes that were used for training and testing. Next, the algorithm was tested on glaucomatous eyes. Both stable and progression was determined by OCT retinal nerve fiber layer measurements 18 months after DARC was taken. The algorithm demonstrated 85.7% sensitivity and 91.7% specificity in these eyes. A significantly higher DARC count was seen in the glaucomatous eyes that were progressing compared to those with stable disease at 18 months. The positive predictive value of DARC was 100% for predicting glaucomatous progression 18 months later. All patients who had a DARC count greater than 30 had experienced glaucomatous progression by RNFL changes on the OCT by 18 months. The number of baseline topographically correspondent abnormal sectors on OCT, RNFL, and Brooks membrane opening minimum rim width imaging significantly increased in patients whose glaucoma progressed versus those whose disease was stable, but this was the only other parameter that showed this difference. In an upcoming glaucoma validation study, we will be evaluating DARC as a measure of latanoprost treatment efficacy, but also for DARC for predicting disease progression at 2 years, and also in a retest assessment of the DARC count. In the meantime, a company called Novi Limited has been developing DARC technology and is now collaborating with multiple pharmaceutical companies to test drug efficacy in glaucoma, amongst other conditions. In the phase 2 clinical trial, we were able to also analyze 29 eyes with macular degeneration. These eyes had DARC only once at baseline, but because patients were being treated usually for wet AMD, they were followed up regularly in clinic, and so we were able to follow up with substantial OCT information. We found that a DARC spot at baseline was predictive of a new area of exudation up to 36 months later. Further, patients with a DARC count greater than 5 at the macula ultimately developed new lesions of wet AMD. Although larger validation studies are required, this finding shows the potential of DARC as a biomarker of wet AMD. We are also investigating the role of DARC in predicting the progression of geographic atrophy. In our phase 2 study, we found that DARC was predictive of the direction of GA growth. Additionally, when we looked at all eyes with growing GA, patients with a DARC count greater than 10 went on to develop larger areas of rapid growth of atrophy. We are currently conducting a study to explore the use of DARC as a predictor of macular atrophy. We are also working with pharmaceutical companies to explore the use of DARC in macular degeneration and diabetic retinopathy, and using it as an exploratory endpoint and a measure of treatment efficacy. Hopefully, more data will allow us to put this forward as a technology that can be used not only as an exploratory endpoint, but ultimately as an approved clinical endpoint. The DARC metrics that we are evaluating include the DARC count, DARC density, DARC spatial map, and DARC morphology and signal. We're also working to develop an intranasal formulation of DARC which would be less invasive for patients and allow rapid entry into the retina and potentially require a reduced dose, thereby reducing cost. If DARC is to be used as a screening test, it must eventually be non-invasive, applicable, sensitive, and specific. Ultimately, DARC could play a role in multiple diseases by helping to measure the impact of a treatment or intervention by assessing disease activity, identify non-responders to existing and new interventions, and also stratify patients in clinical trials and in clinical practice, but in particular to create enriched cohorts consisting of those at the highest risk of disease progression. In several spaces, these benefits will be particularly valuable as the number of patients increases worldwide due to the aging population.
Thank you for tuning into this episode of GT the Podcast. If you have any feedback or topic suggestions, find us on Instagram, LinkedIn, Facebook, or Twitter. And stay tuned for more hot topics in glaucoma care on GT the Podcast.